Okay, so what I decided I would do after Roger's really fantastic talk yesterday that left us all terminally depressed, I thought, but did give a really accurate view of the state of where we are, and to try to. So I told Roger yesterday that I was going to give the talk that he gave. So last night he blew my evening. Instead of going dancing, I ended up completely rewriting my talk as a result. And what I'm going to try to tell you is what some of what we're doing at the NIH to try to address some of the problems that we've been talking about. Certainly not all of them, but certainly some of them. So. Okay, so let's just dive right in here. So this is the problem that we're dealing with. I think the words that you've heard, both of them are right. I mean, somebody said to me this morning, it seems like we're dealing with, one side that says everything's great, we're going in the right direction, amazing capacities, and the other side says, gosh, we're things aren't going so well. And they're both true from our perspective. I mean, on the left side, we have on the upper left there, thank you, Eric, wherever Eric went, there he is. For those of you who don't remember ancient history, that's Lander et al. In the upper left corner, and the bottom is embryonic stem cell or iPS cells differentiating into neurons. And so just amazing things that when I was in training 35 years ago, we just couldn't possibly imagine. And so what's the ability of science to discover things and what it has discovered already has changed utterly in the last 35 years. But if you ask how we're doing on the health side, things really haven't changed that much for the most part, except for a few cancers and increasingly more cancers. But for my own field of neurology, very little has changed during that time. And there's this wonderful IOM report called Shorter Lives, Poorer Health. That's in the upper right corner that kind of talks about that. And then the bottom right is a child with progeria who's about 11 in that picture, and he was dead the year after from cardiovascular disease. It's a premature aging syndrome. And so it's a really interesting question. To the degree that health is based on science, why has health not kept up with science? And one of the reasons for that is that we're just really bad at going from what's on the left side to going on the right side. We just don't know how to do it very well. And so it's as Roger was saying yesterday, it's a failure-prone, inefficient, costly process. And something we haven't talked about, but which is equally true, is that even once an intervention is developed, it takes about the same amount of time, 15 to 20 years, for that intervention to get to all the patients who could benefit. And so while translation is not new, it's been going on for 4,000 years, the opportunity is greater, both because we understand a lot more about disease and we have technologies we didn't before. So the question is, how do we deliver on the promise of what's on the left side for all the people on the right side who are waiting? And this is just a graph I love to show because it encapsulates my career, but it also sort of graphically shows one example of how different things are. So when I graduated from medical school back in 1986, that was before this graph started, and it was easy to study for the medical boards because there were only about 10 human conditions, the molecular basis of which was understood. And now that number is over 6,500. And so, and that's great, and that just fundamentally changes how we can think about these disorders. But from a patient perspective, this is what you really care about, that there's only about 500 of these that have any treatment. So numerically, about 95% of human diseases have no treatment whatsoever, FDA-approved treatment. And most of these, of course, are numerically, they're rare diseases. And when I think about the diseases that I used to treat in the clinic, ALS, Huntington's disease, brain tumors, intractable pain syndromes, these are just as untreatable now as they were before, despite amazing understanding of the genetics and molecular biology of these disorders. So what I learned when I was in clinical training and and research training turned out to not be true. I know that's a shock. that what they taught us in school wasn't the whole story. But this is what I was taught, that, I'm a neurologist, so I was taught, well, all you got to do is make a diagnosis and you're done, right? That's the old saw about neurologists. So that's a stroke in the upper left corner there, and as a geneticist, you do the same thing. That's actually the original paper reporting the original genetic disease. That's Linus Pauling, 1949, sickle cell disease, sickle hemoglobin. And I was sort of taught that, well, gosh, as soon as you have this, the development of the therapy and dissemination of it just happens by itself. It's a thermodynamically favored process, like it says in the paper. Within 3 to 5 years, there will be a drug based on this, right? Well, it turns out not to be true. And this is something actually Roger taught me when I worked with him at Merck 20 years ago. That this does not equal this. And this, by the way, that's a device at the bottom in case you're wondering. This does not equal this. So these inequalities are the translational problem, really. How do we get from the first to the second to the third much more efficiently and effectively than we do now? So this is what we call translation. Everybody uses this term a little bit differently, so we had to actually define, because we have translation in our name, we had to define what we mean by this. And fundamentally, there's a lot of words here, but really it's fundamentally turning an observation into an intervention. The first time a scientist in the lab, a doc in the clinic, a public health worker in the community makes an observation and a light bulb goes on and they say, this mechanism, this cellular phenotype, this clinical observation, this public health problem, if I could just reverse what's in front of me, then I could make the target better, the cell better, the animal better, the person better, the public health problem better. The whole problem from that ideation, the whole process from that ideation to making the physical thing, the drug, the device, the behavioral intervention, and then showing that it is effective in cells, animals, clinical trials, controlled environments, and then what a friend of mine likes to call free-range humans, who is humans in the environment. That whole process is translation from our point of view. Because when you ask, well, if we're going to translate from fundamental understanding to health, where does health happen? Health does not happen in a clinical trial. It happens in the community. So until we have shown that the original idea actually works in a public health setting, we can't claim victory. So that's our view of this. But you notice that we're not the National Center for Translation. And this is something that somebody was talking about yesterday. The original idea of the center that I run was that it was going to make drugs and it was going to, teach Pfizer and Merck and Novartis how to make drugs, which is one of the more absurd things that's been said ever. And that's really not the point. The point is translational science. What is translational science? Translational science is like any other science, but it's a new science. It's the field of investigation that seeks to understand what the general principles are underlying each step of the translational process, as I just described it. The translational process, for the most part, is a relentlessly empirical trial and error process. And if you have a process that has this many steps, 20 or so steps, going from the beginning to the end, as I just described it, with an increasing number of degrees of freedom and an individual success rate yield at each step at about 50, 30, 20%, 10%, you multiply all that, then the likelihood of your success, you're getting to the end, is say 0.3 to the 20, right, which is a very small number, which is about what you see, going from a target to a public health intervention. So the way to improve that is not doubling the number of projects you do, not shots on goal, because then you go from 1 times 10 to the minus 5 to 2 times 10 to the minus 5. The answer to this is to think about it like a complex biosynthetic reaction with rate-limiting steps. You develop catalysts to those rate-limiting steps, and that will increase your yield by orders of magnitude like any other catalyst does. So that's what NCATS does. Okay, so this is our mission. Doesn't matter too much for this purpose. I just use this as an introduction to the next slide. Our purpose is to develop new ways of doing things, new methods, new technologies that will make this whole process better, faster, cheaper. And so we have a lot of the people and technologies and tools and thinking that exist in biotech, pharma, et cetera, as well as in academia. But we can do things that were impossible for me and my colleagues when we were in industry because we don't have a short-term commercial imperative. You know, our product is knowledge, not a product, which is really liberating and quite a privilege. So these are some of the problems that we work on, and a number of them have been talked about today or yesterday. These are the problems that bedevil every translational project. It doesn't matter whether you're working on what disease you're working on, you know, Alzheimer's disease or psoriasis or Menkes disease, they're all the same problems. They've never been the subject of the scientific method before. How do we understand how to do each of these better? How do we develop interventions to make them better and measure whether we're doing better in each of them? And you could ask, well, why has that, if there's such big problems, why haven't they been worked on before? It's really because it hasn't been anybody's day job. You know, these fit into what have been called the tragedy of the commons. You know, these are everybody's problem in general and nobody's problem in particular, so nobody solves them. You know, they're like clean air or clean water. They are NCATS' problem. You know, sometimes I think about, you know, NIH is full of institutes with disease names, the National Cancer Institute, arthritis, musculoskeletal diseases. Our disease, if you will, is the system by which to do translation and setting that on a firm scientific footing. So this is the way we think about it. We often talk about translation as a linear process, but of course it isn't and never has been. Up until the last 50 years, it has been impossible to start with a reductionist approach, start with a protein or a gene, and go forward to a public health observation because it was impossible to isolate a protein or a gene. Before cloning was possible, you couldn't start out with a clone gene. So for the last 4,000 years, translation has started with public health observations. I mean, think about John Snow and the water pump. That's where it started where this kind of observation started, and the identification of the etiologic agent being Vibrio cholera for cholera. That is starting with public health at the top and going counterclockwise. Now we can start with basic research and go clockwise too. But the idea is that they can go clockwise or counterclockwise. And you notice right in the middle, we put patients right in the middle. Interestingly, you know, even though what we do we think about it as a consumer good, really. You know, it's things we hope people will use. They're fundamentally no different than toothpaste and clothing. Hope maybe a little bit more important, but fundamentally it is. And yet we have not involved the hoped for consumers in that process the way we do with any other consumer good. And we think that's an unforced error, and that doing that will actually improve the urgency and relevance of the work we do. And actually we have some data supporting that now. So the first thing we did, and one of the first things we did, is to deal with the linguistic problem. And I don't have time to talk about all the linguistic things that we're doing, but there are many linguistic problems in this space because of the different languages that, say, physicians and geneticists and pathologists and cell biologists, et cetera, use. But the first thing we dealt with is this word pipeline. So we talk about drug development pipeline. Well, and this is this chevron diagram you've probably seen before, start with basic, end up in FDA approval or post-marketing. A pipeline is a conduit into which an entity starts at the beginning and comes out the end unchanged, and the progress through that process is dependent only on the diameter of the pipe. That's a pipeline. Now that is about as inaccurate a description of translation or drug development as there could possibly be, and yet I've used the term from the beginning, and guess what? The public believes us. And so they don't understand, if this is a pipeline, what's the matter with you people? You can't get water to go through a pipe? So we realized we really needed to change our terminology. So through a broad public-private work through the National Academy of Medicine, we have converted this from this chevron into this. And this is actually what it looks like. And as soon as you show this to somebody, they say, oops, because my basic science colleagues are all up in the upper left corner. You know, and that's what I am. I'm a modelism geneticist by training. and they realize this is not a pipeline. This is the most horrific board game you've ever played in your life. And so we're using this not only for educational purposes, but also to help people orient themselves on this map. We're actually creating a Google Map version, an interactive version where you can zoom in and zoom out, have a GPS function to identify yourself of where you are, and then like on a Google Map, you can link out to see, if I want to go get a cup of coffee someplace, where is the nearest place to get a cup of coffee? Once you identify where you are on the map, to link out to programs at various places, which will help you from getting from point A to point B. So something about personalized medicine I just wanted to sort of set up here for a second. We do a lot of work in rare diseases at NCATS, and we do it for essentially two reasons. One is that they really are the subject of disproportionate suffering and death in pediatric populations, and I'll get to that in a second. But the other is that they are, for want of a better term, they are the model organisms of the translational space. If you're going to try to figure out how to do this better, faster, cheaper, don't do it in a complex disorder. Like if you're trying to figure out genetics, don't do it in a human, do it in a yeast. And what we learn in a yeast, we hope will be, not always, but we hope it will be applicable to humans. And this is the same sort of idea. So what we've realized in doing this is listening to particularly the most recent, say 5 to 10 years discussion of personalized medicine. Sometimes it's talked about as if it's new, and these topics are things which the medical field has never dealt with. In fact, personalized medicine is really the upside down rare disease medicine. That is, and in some ways is the same. So if you look at what we're looking at here, is a complicated graph, but if you look at prevalence going from high to low on the left side, and cost going from low to high on the right side. If you start out with a common disease, which is in the US is greater than 200,000 people, and then you go from that to a rare disease, which is less than 200,000, to a personal disease, let's call it one person. And you look for examples. Well, lung cancer is a great example of a common disease. The treatment for that might be cisplatin, $20 a dose or so. As you get less common, look at ALK positive, ALK positive lung cancer. Crizotinib's a great drug. It costs about $15,000 a dose, not $20 a dose. As you get to a less common disease, Hunter syndrome, MPS II, there's an enzyme replacement. It's about $400,000 a year per patient for life. As you go down to a personal disease, whether it's CAR T or something else, we're in $700,000 to $1 million each. And a lot of the problems that we deal with, not only in cost, but also in how to find patients and the small population clinical trials, et cetera, that are thought to will be a problem in personalized medicine, are already the case in rare diseases. They have been forever. So we can learn a lot about personalized medicine by studying rare diseases. Okay. So a way I've thought about this is going from really what I see, and it's happened in my own career, is in the evolution of how doctors we hope they're going to practice. It's there's this other term called evidence-based medicine, which always scares people. If you talk to a patient and you say you're doing evidence-based medicine, and they say, well, what were you doing before? And really, it was experience-based medicine. That it was every practitioner saw a small subset of the entire population of a given disorder. Some disorders they never saw at all, and then based on that syncretic, individualized, integral of their experience, they treated patients differently. And that's actually worked reasonably well, but it's the shaman model or the artisan model, the Michelangelo model of medical practice. So in this case, you have a patient population that's divided by practitioners, and each of those practitioners subdivides, they see a different part of the population, and so they generate different kinds of data on the same patient population. It's a random distribution of the patient population who just happens to walk into their office, and then based on that, they're going to get 3 different treatments. And granted, doctors sometimes read the literature, but most of the time they don't, actually. Most primary care docs don't, and they just don't have time. And so if you look across geographic distributions, there's huge differences in how individual patients are treated, and that shouldn't be a surprise. But here, the hope is, with evidence-based, personalized medicine, you have the whole patient population, and you generate data on that whole population. That's the idea. And then each of the patients has the data that is applicable to them identified out of this giant data set, and then those data lead to evidence-based personalized treatment. So it's personalization on the basis of the patient, not just the practitioner. And that's one of the ways that I think about this evolution. Okay, so getting into rare diseases for a second, because as my title said, this is sort of what I decided to do with this talk as a view into personalized medicine, and you'll see why I'll say that. So there's about 7,000 of these. About 80% of them are genetic, things you've heard of: sickle cell disease, Huntington's disease, cystic fibrosis, and some you probably haven't heard of, Hallerworden-Spatz disease, and other great sounding names. About 50-60% of them have onset in childhood, and an interesting number is that there are about 250 new ones discovered every year. And that number is not plateauing. It's really interesting. New syndromes or new phenotype-genotype correlations. They're individually rare by definition, but they're cumulatively common because there's so many of these. The total population prevalence is about 8% of the population. It's about the same as type 2 diabetes. But I bet you hadn't thought about it that way. Very high direct patient costs and loss of productivity, and this was both in the patient and the family. This was referred to yesterday. Interestingly, for a variety of reasons, accurate diagnosis takes between 5 to 15 years. It's known as the diagnostic odyssey. And during this time, huge healthcare costs in the patient's quality and quantity of life goes down during this time. And as we said, only about 5% of these have any improved treatment, about 400 of these. And I did a couple of calculations a few years ago which were illuminating. One is that if we had a drug for every rare disease patient in the US today, and we priced it at a rather modest level, $200,000 a year, which is really a bargain in rare diseases now, that drugs for rare disease patients alone, forget the common ones, rare disease patient drug alone would consume 1/2 the US GDP. 1/2 the US GDP just for rare disease drugs. So in other words, don't bother. Ain't gonna happen. Then I did another calculation. At the current rate, we don't have to worry about this too soon, because at the current rate of progress, how many years will it be before there is a disease, before there is a treatment for every disease, an FDA-approved treatment? The answer is 2,000 years. And let me tell you, if you talk to a parent of a child with a rare disease and you tell them it's going to be 2,000 years before we get to you, it doesn't go over well, and I would argue that it doesn't have to go over well. Back when I was in training 35 years ago, we didn't know what caused these things. So there was no way we could even get a hold of this problem. I think now we do have a way to get a hold of this problem, but we're going to have to do things a little differently to realize, to change this course. So one of the things that we're doing, and to bring us back from the abyss, and whenever I give this talk, it's a little bit like Roger's thing, oh my God, you're depressing us. There are solutions to this, and you heard some of them yesterday. One of the things that we're doing is transitioning from the one disease at a time to the many diseases at a time, looking at this disease, what's common among them, not what's different. That's different from how NIH and most researchers work up until now for reasons that you know, but also platform technologies that could improve diagnosis and treatment. So we're doing this in a number of ways, providing information to patients, big data like I just talked about, empowering patients as research partners. We think this is a real disruptive technology which can really improve the efficiency in this space. Interoperable data registries, doing a lot of natural history, interventional studies across a very, very broad rare disease clinical research network. We do a lot of collaborative development of treatments with academics and biotech and pharma companies. One of the things that we're working on now is development of gene therapy platforms, so people don't have to compete on the basis of the vector. They can compete on the basis of the insert. If you think about in gene therapy, a lot of the time, what companies are doing is tweaking a vector a little bit so they can get IP on the vector, and then that allows them to sell what happens to be the insert in the vector for a very large amount of money. And just imagine you had 2 or 3 or 5 vectors which had gone through all the tox studies, all the CMC manufacturing studies that the FDA requires, and then all an investigator would have to do is swap in whatever the insert was. That would dramatically lower both the barrier to entry and the cost of these kinds of technologies. And this is completely doable. It's just that the only people who are going to do this are folks who don't have to get a return on investment. And so that's one of the things we're doing, and focusing on the very, very rare diseases. Of the 7,000 diseases, it's a real long tail, so many of these are 1 in a million, 1 in 100,000. They're very, very, it's really unclear, how you would ever make money on that low of prevalence, except by charging a very, very, very large amount of money, which, as has been said before, is not sustainable. I did want, because nobody had done this so far, and I think we all think this is really important to do, to remind ourselves of why we're doing this. So this just happens to be a demonstration case that we did of a novel platform with a company called Agilus, which is in Boston, and some investigators in Taiwan, and then eventually was acquired by PTC Therapeutics. But this happens to be on a rare pediatric disease called AADC deficiency, and this is a child who's 4 years old in this picture. As a neurologist, these are the kids I used to see. You notice she's not reaching out. She's not responding to her family. When her father raises her arms, she sort of grimaces, but it's hard to know whether it's purposeful or not. And she would have gone downhill from here. A year after gene therapy, which in this case was the ADCD gene into the putamen, this is the child. Now, when this happened, it was formally possible that this is a spontaneous remission, but this has never been seen before in the history of this disorder. And now there's dozens of kids to whom this has happened. And if you go a year later, so this is 2 years from the one on the left, she's now in kindergarten. And if you watch this video, it's very hard to distinguish her from her classmates. Now, there's no reason this is a special case, right? So the question is, if we can do this, don't we have to pull out all the stops to figure out how to do this more broadly? And initially in rare diseases, and then apply what we're doing, granted much, much, much more difficult in the more common diseases. Okay. And something that, if you haven't caught up with this is a really interesting story. This happens to be a variant of a particular version of a disease called Batten disease, caused by a mutation, rare mutation in a gene called CLN7. This mom and her daughter Mila in the middle it drove the development of a personalized oligonucleotide therapy that resulted in a splice change, which is what the difficulty is in one of her alleles. The investigator on the left, Tim Yu, who's at Children's in Boston, did this with them, and they took the model that had been developed for Spinraza, which is the oligo treatment for spinal muscular atrophy developed by Biogen, and said, well, gosh, if we just use the same oligo chemistry and we inject this like Spinraza was into the cerebrospinal fluid, could we have a very rapid development of this oligonucleotide from identification of this girl's mutation to a treatment in less than a year? And in fact, that's what they did. And so we're now working with them to ask, and the FDA, if we wanted to do this for all, there's about 1,000 other diseases that fit this molecular phenotype of a splice problem, and we worked with FDA.
Try, please, to keep the time,
yeah.
Everybody.
And we worked with FDA to say, what would we need to do, to make this workable? That's what we're doing now. So finally, what are we doing in this problem of why drugs fail in development? To a great degree, it's because rats are not humans. Humans are not rats, and what works in a rat is not necessarily work in a human. We've known that for a long time, but what's been difficult is to figure out a better way to do it. So just to give you a sense of what we're doing in this space, in this very large collaboration between pharma and us and the FDA and DARPA and lots of other folks, we have developed over the last 7 or 8 years microphysiologic systems. That is, these are microfluidic-based mini tissues which represent the structure and function of all human tissues, and we're using those to test for safety and efficacy rather than animals. And it turns out, for a variety of reasons, to be really a transformational technology. And so we're doing this, we're now using these to model rare diseases, and because you can do these, you can develop these based on induced pluripotent stem cells, you can look at variation in the population as well for both rare and common diseases in ways that were just impossible before. We do a lot of patient-driven drug development as well. Roger, I don't know if you recognize that gentleman in the lower right there. That's Jim Inglaze, who used to work at North Wales and now works at our place. So these are patient-driven programs, patients that came to us and we work with them. We also work with patients on the distal end. This is completely the opposite end. This is the problem of, well, gosh, if you have a treatment and nobody takes it, well, so what? So this is a problem where we made a collaboration with barbers. And you might say, well, you know, barbers on the research team, that's pretty novel. And it turns out that barbers are trusted in the black community, and young men need to go get their hair cut, and they don't like to go to the doctor. But if you put pharmacists in a barbershop and you have the pharmacists talk to their clients while they're getting their hair cut, you can get them on blood pressure medicine and get them treated. So the promise of personalized medicine is seen through rare diseases. You can get precise diagnosis, often with implications for care, even if you don't have any regulatory approved treatment. Treatments often have large clinical and functional effects, as others were saying yesterday, and that's really wonderful if you can identify these. And it's subset common disease, same thing, ALK positive lung cancer is an example. Involvement of patients and family members, which this brings about, really does increase relevance, urgency, recruitment, adherence, all those things. Paradoxically, we found, not always, but we found in the general case, the sociology of this is that this research has led to community efforts among rare disease patients to share data, develop interoperable registries. There's this community mindedness that really can be transformative in this community, which was a little it was unexpected, at least by me. But what are the perils? Well,
we try to keep time, sorry for
this is the last one. So the perils, high cost. Let's just leave it at that. Some interventions are even unworkable in the target population, like sickle cell disease. We have had this experience, and others have, that involvement of patients can actually be counterproductive at times. It can lead to misunderstandings which can slow progress. Just as an example, I made the mistake of teaching a rare disease father the word equipoise. And it's like giving Prometheus fire, you know, and so we're sitting in an FDA meeting, and he says to me and the FDA people, I decide when equipoise has been reached for my child, not you. Interesting. It's not currently applicable to diseases that have major morbidity and mortality. We talked about this yesterday. And something that we are working on, but and this group should be interested in as well, the patients in developing countries really get left behind because of the more common diseases that really garner their attention, not inappropriately. So I hope that's given us some food for thought, and hopefully we can talk about this as the day goes on.
Thank you very much. We'll keep the questions to start in the session concluding the morning, and